Welcome back, class, to AGR2 IPM. In this lecture, we'll be talking about control techniques relating to biotechnology and genetic engineering. Let's start by considering traditional plant breeding. So for thousands of years, human selection for features such as faster growth, larger seeds and sweeter fruits has dramatically changed domesticated plant species compared to their wild relatives. Remarkably, many of our modern crops were developed by people who lacked an understanding of the scientific basis of plant breeding. Here we have a carving from 870 BC showing the artificial pollination of date palms. Despite being poorly understood, plant breeding was a popular activity. Gregor Mendel, the father of modern-day genetics, was a plant breeder, as were some of the leading botanists of his time. His 1865 paper explaining how dominant and recessive alleles could produce the traits that we see and could be passed to offspring was the first major insight into the science behind the art. This was ignored at the time, but rediscovered in 1900 when three scientists working on breeding problems rediscovered Mendel's paper and publicised these findings. Breeders then brought their new understanding of genetics to the traditional techniques of self-pollinating and cross-pollinating plants. Breeders of corn tried numerous strategies to capitalise on the insights into heredity. Corn plants that had traditionally been allowed to cross-pollinate freely were then artificially self-pollinated for generations and crossed to other self-pollinated lines in an effort to achieve a favourable combination of alleles. The corn that we eat today, of course, is the result of decades of this breeding strategy of self-pollination followed by cross-pollination. So the art of recognising valuable traits and incorporating them into future generations of plants is very important in plant breeding. Breeders have traditionally scrutinised their fields and travelled to foreign countries searching for individual plants that exhibit desirable traits. Sometimes these traits arise spontaneously through a process called mutation, but the natural rate of mutation is too slow and unreliable to produce all the plants with favourable combinations of alleles that breeders would like to see. In the late 1920s, researchers discovered that they could greatly increase the number of these variations or mutations by exposing plants to X-rays. This mutation breeding accelerated after World War II when the techniques of the nuclear age became widely available. Plants were exposed to gamma rays, protons, neutrons, alpha particles, beta particles to see if these would induce useful mutations. Chemicals too were used, such as sodium azide and ethyl methane sulfonate, in order to cause mutations. Quite a few cultivars have been developed via mutation breeding, as you can see listed here. Mutation breeding was particularly popular in the United States during the 1970s. And although interest has waned somewhat in recent years, occasional varieties continue to be produced using these methods. For example, the new or newish herbicide-resistant wheat variety called Above that you can see in this table was developed using exposure to sodium azide. And mutation breeding efforts continue around the world today. Another method for increasing the number of mutations in plants is tissue culture, often performed in small glass or plastic containers. Tissue culture was not actually developed with the intention of causing mutations, but the discovery that plant cells and tissues grown in tissue culture would mutate rapidly increased the range of methods available for mutation breeding. Haploid breeding. In the 1920s, spontaneously occurring haploid plants were discovered. Haploid plants have half the number of chromosomes. But haploid breeding was not a practical technique until methods for the controlled production of haploid plants were developed. Once a haploid plant has been obtained, its chromosomes are artificially doubled using a chemical called colchicine, and this returns the plant to the normal number of chromosomes. In other words, a diploid plant is restored. And this type of plant is valuable because the chromosomes that were created by the artificial doubling are exact copies of the chromosomes that were originally present in the haploid plant. In other words, the plants are 100% homozygous at every locus. Haploid plants have been used in creating cultivars of barley, maize, tobacco, asparagus, strawberries, and tall fescue grass. But often these plants are more useful in basic research than in commercial applications. While most breeders cross-pollinate plants of a single species, some breeding methods rely on crosses that can be made between two species within the same genus. So, for example, a cross between two different species of Musa produced the sterile banana plants with which we're familiar with. Less commonly, the cross is between two members of two different genera. For example, a cross between wheat and rye produced the grain called triticale, which contains a copy of all the chromosomes from both species. A variation on this crossing procedure is to select plants that have single chromosomes or chromosome regions, which we call arms, substituted from one species into another. Many of today's modern wheat cultivars, for example, contain a chromosome arm from rye, which adds resistance to several diseases. Transgenic biotechnology. This provides the means to make even more distant crosses than were previously possible. So organisms that have until now been completely outside the realm of possibility as gene donors can be used now to donate desirable traits to crop plants. These organisms don't provide their complete set of genes, but rather they donate only one or a few genes to the recipient plant. So, for example, a single insect resistance gene from the bacterium Bacillus thuringiensis can be transferred to a corn plant to make Bt corn. Transgenic plants were first created in the early 1980s by 4 groups working independently. On the same day in January 1983, the first 3 groups announced at a conference in Miami, Florida, that they had inserted bacterial genes into plants. The 4th group announced at a conference in LA, California, in April 1983 that they had inserted a plant gene from one species into another species. The Washington University group had produced cells of Nicotiana that were resistant to the antibiotic kanamycin. The Belgium group had produced tobacco plants that were resistant to kanamycin and to methotrexate, which is a drug used to treat cancer and rheumatoid arthritis. The group at Monsanto Company had produced petunia plants that were resistant to kanamycin, and the Wisconsin group had inserted a bean gene into a sunflower plant. So a transgenic plant contains a gene or genes which have been artificially inserted instead of the plant acquiring them through pollination. The inserted gene sequence is called the transgene, and this may come from any other organism. And this is based on the concept that all living organisms share the same genetic code. So theoretically, any gene should be able to function in any organism. But clearly the situation is not that simple, because even after genes have been identified, cloned, and then added to the genome of a new plant, the genes may be modified by the location that they end up in within the genome. They might be modified by the presence of other genes and even by the environment in which the organism lives. So plants that contain transgene are referred to as genetically modified crops or genetically modified organisms, GMOs, although in reality all crops have been genetically modified from their original state by domestication, selection and controlled breeding over long periods of time. So why make transgenic crop plants? Combining the best genes in one plant is a long and difficult process, especially because traditional plant breeding has been limited to artificially crossing plants within the same species or with closely related species in order to bring the different genes together. So, for example, a gene encoding a protein in soybean could not be transferred to a completely different crop such as corn using just traditional techniques. Transgenic technology, on the other hand, enables plant breeders to bring together in one plant useful genes from a wide range of living sources, not just within the crop species or from closely related plants. So this technology provides the means for identifying and isolating genes controlling specific characteristics in one kind of organism and for moving copies of those genes into another different organism, which will then also have those characteristics. So this is just another tool that enables plant breeders to do what they have always done, that is, generate more useful and productive crop varieties containing new combinations of genes. But it also expands the possibilities beyond the limitations imposed by traditional cross-pollination and selection techniques. Desirable genes may involve higher yield, improved quality, pest or disease resistance, and tolerance to abiotic stresses such as heat, cold and drought. Let's take a brief look at designing transgenes. So once a gene has been isolated and cloned into a bacterial vector, such as a plasmid, it has to undergo several modifications before it can be inserted into a plant. So firstly, a promoter sequence must be added for the gene to be correctly expressed. That is translated into a protein product. The promoter sequence it's like an on-off switch that controls when and where in the plant the gene will be expressed. To date, most promoters in transgenic crop varieties have been constitutive promoters. That is, they cause the gene to be expressed or switched on throughout the life cycle of the plant in most tissues. The most commonly used constitutive promoter is the CAMV35S promoter, or the cauliflower mosaic virus promoter, and this generally results in a high degree of expression in plants. Other promoters are more specific and they respond to cues in the plant's internal or external environment. An example of a light inducible promoter is the promoter from the CAB gene, and this promoter encodes, the CAB gene encodes the major chlorophyll binding protein. So you can see in this image the promoter in yellow, which is placed just before the start of the transgene, shown here in blue. Sometimes the cloned gene is modified to achieve greater expression in a plant. So, for example, the Bt gene for insect resistance comes from bacteria, and it has a higher percentage of adenine thymine nucleotide pairs compared to what you would find in plants, which prefer guanine cytosine nucleotide pairs. So in a clever modification, researchers substituted some of the AT nucleotides with GC nucleotides in the Bt gene without significantly changing the amino acid sequence. And the result was enhanced production of the gene product in plant cells. A termination sequence is also added, and you can see that in the figure here in red. This termination sequence signals to the cell machinery that the end of the gene sequence has been reached. In addition, a selectable marker gene, shown in green, is added to the gene construct in order to identify plant cells or plant tissues that have successfully integrated the transgene. And this is necessary because achieving this incorporation and expression of transgenes in plant cells is actually quite a rare event, and it occurs in just a few percent of the targeted tissues or cells. So it's not a very efficient process. So these selectable marker genes encode proteins that provide resistance to agents that are normally toxic to plants, such as antibiotics or herbicides. So only plant cells that have integrated the selectable marker gene as part of this transgene construct will actually survive when grown on a medium that contains the appropriate antibiotic or herbicide. As for other inserted genes, marker genes also require their own promoter and termination sequences for proper function. Transformation is the heritable change in a cell or organism brought about by the uptake of introduced DNA. So transforming plants utilises mostly one of two different methods. First, the gene gun method, otherwise known as microprojectile bombardment or biolistics. Secondly, the agrobacterium mediated transformation method. Biolistic transformation using a gene gun bombards protoplasts, that is plant cells with the cell wall removed, with tiny metal particles coated with the foreign DNA. The metal particles are often gold or tungsten. Agrobacterium-mediated transformation utilises a bacterium called Agrobacterium tumefaciens. This is a soil-dwelling bacteria that has the ability to infect plant cells with its own DNA. It causes crown gall disease in many ornamental and fruit plants. Transformation using Agrobacterium has been successfully practiced in dicots, such as soybeans and tomatoes, for many years now, but only recently has it been effective in monocots, that is grasses and their relatives. In general, the agrobacterium mediated transformation method is considered preferable to the gene gun because of the greater frequency of single site insertions of the foreign DNA. This makes it easier to monitor. The DNA in an Agrobacterium tumefaciens cell is contained in the bacterial chromosome, as well as in another structure known as a TI, or tumour-inducing plasmid. The TI plasmid contains a stretch of DNA termed tDNA. It's about 20 kilobases in length, and this tDNA is the region that's transferred to the plant cell during the infection process. A series of genes within the tDNA are referred to as virulence genes, and these genes direct that infection process. Agrobacterium tumefaciens can only infect a plant through wounds. When a plant root or stem is wounded, it gives off certain chemical signals. In response to those signals, the virulence genes of Agrobacterium become activated, and they direct a series of events that are necessary for the transfer of the tDNA from the TI plasmid into the plant's chromosome. So this is briefly how it works. Different virulence genes will copy the tDNA. They'll attach a product to the copied tDNA strand. They add proteins along the length of the tDNA, possibly as a protective mechanism. They open a channel in the bacterial cell membrane through which the tDNA passes. The tDNA then enters the plant cell through the wound. Now, it's not clear how the bacterial DNA moves from the cytoplasm to the nucleus of the plant cell, nor is it clear how the tDNA then becomes integrated into the plant chromosome. So remember that most of the time plant DNA doesn't exist as exposed strands of DNA, but it's wrapped with or wrapped around histone proteins. One speculation is that the tDNA waits until the plant DNA is being replicated or transcribed, and then it inserts itself into the exposed plant DNA. So to harness Agrobacterium tumefaciens, scientists have removed the tumour-inducing section of the tDNA, and this is where the transgene is inserted into this region. Following the gene insertion process into the plant tissues or plant cells, plant tissues are then transferred to a selective medium which contains an antibiotic or herbicide, depending on which selectable marker was used. Then only plants expressing that selectable marker gene will survive, as shown in this figure, and it's assumed that these plants will also possess the transgene of interest. Thus, any subsequent steps in the process will only use these surviving plants. Transgenic plants are then regenerated under controlled environmental conditions in tissue culture. Intrinsic to the production of transgenic plants is an extensive evaluation process which verifies whether the inserted gene has been stably incorporated without detrimental effects to other plant functions or product quality or the intended agroecosystem. Initial evaluation includes attention to activity of the introduced gene, stable inheritance of the gene, and unintended effects on plant growth, yield and quality. If a plant passes these tests, most likely it won't be used directly for crop production, but it will be crossed with improved varieties of the crop. And this is because only a few varieties of any given crop can be efficiently transformed. And these generally don't possess all the producer and consumer qualities that are required of modern cultivars. The initial cross to the improved variety has to be followed by several cycles of repeated crosses to the improved parent, a process known as backcrossing. The goal here is to recover as much of the improved parent's genome as possible with the addition of the transgene from the transformed parent. The next step in the process involves evaluation trials in greenhouse and in the field to test the effects of the transgene and also overall performance. And this phase also includes evaluation on environmental effects and food safety. The most important transgenic crop in terms of acreage planted is soybean. followed by corn, cotton and canola. Adoption of transgenic crops in the United States has been far greater than in any other country. This table summarises the transgenic crop production area by country as of 2000. Most of the transgenic crop varieties currently grown by farmers are either herbicide tolerant or insect pest resistant. And in addition to these crops listed here, minor acreages have been planted to transgenic potato, squash and papaya. This table summarises worldwide production area of transgenic crops as of 99, and also some of the traits are listed here. For example, herbicide tolerance, Bt insect resistance and virus resistance. Of course, most of you will be familiar with Bt insect resistant crops from previous subjects that you've studied. Bt is short for Bacillus thuringiensis, a soil bacterium whose spores contain the cry protein. In the insect gut, this protein breaks down to release a toxin which binds to and creates pores in the intestinal lining. This results in ion imbalance, paralysis of the digestive system, and subsequent insect death. Different versions of the cry genes, in other words the Bt genes, have been identified, and they're effective against different orders of insects. More than 30 subspecies of Bt have been identified, and many of these are registered products used here in Australia, in the USA, France, Germany, Russia, India and elsewhere. 3 products containing Bt, only 3 products of several are listed here. Dipel, which is sold as an organic insecticide. It contains live spores and toxins produced by a subspecies of Bt, that is subspecies Kerstaki. It's used to control various caterpillars in grapes, kiwi, fruit, tomatoes, and many crop species. Fungicide is very similar, also used to control various caterpillar pests. Vectobac, this contains spores and toxins of Bacillus thuringiensis subspecies israelensis, and it's used to control several species of mosquito larvae. These products are considered safe for mammals and birds, and they're safer for those non-target insect beneficials than conventional insecticide products. The European corn borer, shown on the left, and the cotton bollworm on the right are just 2 pests controlled by Bt corn and Bt cotton. The top photo shows Bt corn to the left and a hybrid corn which is susceptible to European corn borer on the right. You can clearly see the difference here. In the photo to the bottom of screen, we can see the results of insect infestation on non-Bt cotton to the left and Bt to the right. Let's consider now the concept of resistance, particularly when it comes to Bt hybrid varieties. Corn rootworm is a serious pest of corn in the US, and it damages roots of seedlings. It's responsible for the application of the largest amount of insecticide in US cornfields. It's applied directly to soil, it leaves residues and leaches into groundwater. Now, although Bt rootworm protected hybrids offer pest management and environmental benefits, there are serious concerns about the development of resistance to Bt from this adaptable insect. In the photo to the bottom right of screen, we can see a corn rootworm feeding on a young corn root. The image to the left shows the range of damage due to corn rootworm feeding, from severe damage to the left through to no damage on the right. So a major concern is that the widespread use of these Bt varieties will accelerate the development of resistance. Measures that are being taken to reduce that risk include a combination of high dose of Bt toxin and also the planting of refuges, where a refuge is an area planted to a non-Bt variety that is physically close to a Bt variety planted field. Beginning in 2000, the EPA required that farmers growing Bt corn must plant at least 20% of their total corn acreage to a non-Bt variety, as you can see pictured here. And the rationale behind this is that the few Bt resistant insects surviving in the Bt field would likely mate with susceptible individuals that have matured in the non-Bt refuge. Thus, the insect genes for resistance to Bt would be swamped by the susceptible alleles. Whether this strategy will work or not still remains to be seen. So in this image that you can see of a Bt refuge strategy, in which at least 20% of the farm's acreage is planted to the non-Bt corn, the capital R refers to the resistant European corn borer adult, and the S refers to a susceptible adult. Let's now revisit the story of the papaya ring spot virus. The papaya ring spot virus, abbreviated to PRSV, infects and damages papaya trees. On the lower right, we can see an infected papaya fruit, and you can easily see how the virus got its name. This virus devastates papaya trees. Here's a healthy plant in the middle of screen, and on the upper left is an infected plant. Here's the culprit. The picture on the upper right of screen is an electron micrograph of the virus, which is obviously very, very tiny. And on the lower left of screen is the kind of insect that carries it around. It sucks up virus particles from infected plants and spreads them to healthy ones. It's extremely difficult to control, and farmers are basically reduced to destroying infected trees. PRSV was first discovered in Hawaii in the 1940s. Within a decade, it had wiped out the papaya industry on one of the islands of Hawaii. The industry then moved to the big island of Hawaii, and it was kept relatively free of the virus for several decades by constant vigilance and rapid elimination of infected trees. Enter the hero of this story, a plant pathologist by the name of Dennis Gonsalves. He heard that a colleague in St. Louis had used the kinds of molecular techniques which we've illustrated earlier to introduce a viral gene into tomatoes, and found that it protected the tomatoes from infection. Gonsalves had worked for many years to derive PRSV-resistant papaya varieties, but was never successful using traditional techniques that plant breeders use, and neither had anyone else in the world. So he decided to try this new molecular approach. But keep in mind that papaya are not easy to transform with Agrobacterium tumefaciens. But he persuaded a colleague of his, Dr. Sanford, to try using his newly invented gadget that he called a gene gun to introduce a viral gene into papaya plants. The so-called gene gun, as you already know, shoots tiny, tiny particles of DNA coated gold or tungsten through the plant cell membrane or wall into the cells. Once inside the cells, some of these DNA molecules will enter the nucleus and become incorporated into the plant's own DNA. It wasn't easy. It took them more than a decade, but the first virus-resistant plants were ready in 1991, and by 92 they'd received permission to carry out the first field trials. At about this time, the virus was beginning to get out of control, and it was causing a lot of damage to the papaya plantations on the Big Island. The picture here at the top left of screen shows you what an infected field looks like. A couple of years later, the USDA gave them permission to do large-scale field tests, which you can see in the bottom, in the image to the bottom right of the screen. But then it took another 3 years to get through all the safety testing required by the USDA, the EPA, and the FDA. By that time, many farmers had simply gone out of business. Fortunately, the story has a good ending. The USDA had allowed them to grow seeds, although they could not give them out until the regulatory requirements were satisfied. The seeds were finally released in 1998, free of charge to the growers, and by the year 2000, the Hawaiian papaya industry had come back to pre-1995 production levels. This is quite a remarkable story, and in many ways is very similar to the history of vaccination in medicine. And indeed, carrying the viral gene essentially vaccinates the papaya plant against the virus, but only by a very different mechanism, which has only been understood recently. Let's have a look at how this works. A virus, as you know, is simply a packet of genes surrounded by a protein coat. Coat proteins are represented on this slide here by the green ovals. The first thing that happens when a virus is delivered into a plant cell by an insect is that the coat proteins are destroyed by the plant defense mechanisms, and we can see that in 1. This releases the genes which commandeer the cell machinery, reprogramming it to make lots more copies of the genes themselves, represented by the wavy bars. These then make lots of new coat proteins that then coat the viral genes to make new virus particles. Then they just wait to be picked up by a new insect in order to infect other plants. Meanwhile, they've pretty much wrecked the cells in which they replicate, and this eventually kills the whole tree. In transgenic resistant plants, the first step is the same. The virus takes off its coat protein. But in the transgenic plants, the invading viral genes are recognised and destroyed, preventing the virus from reproducing. So is this something that happens in nature? Well, yes, it is. This is exactly how plants become immune to viral infection. Very similar to how you become immune to a disease once you've already had it. So the next time that your immune system sees the same virus, your immune system recognises it and destroys it. And this is how it is with plants. Once they've been infected by a virus, this mechanism allows them to resist infection. So what Gonsalves and his colleagues did then was to figure out how to make the papaya trees immune to the ring spot virus by expressing just a little piece of the virus's genetic information in the plant to make it think that it had been infected, even though it hadn't been. He's considered a hero by the local farmers and has received national and international awards for his work. But of course, he's also been attacked and vilified for this work by people and groups who are anti-GMO. Future transgenic crops. First here we have 2 tomato plants artificially inoculated with the bacterial speck disease. The plant on the left has been genetically engineered with a gene for resistance to the disease. Plant on the right is a susceptible non-engineered variety. Okay, additional resistance traits that are also being pursued includes resistance to the disease, the white mold disease caused by the fungus sclerotinia, resistance to the pest Argentina lupa, and also a herbicide resistance trait. These are all being pursued in sunflower. Risks and concerns. The issue surrounding objections to transgenic crops or GMOs in general can be broadly grouped. The first group here involves issues relating to human health: allergenicity, horizontal gene transfer, antibiotic resistance, eating foreign DNA, cauliflower mosaic virus promoter, and changed nutrient levels. Let's look at each of those in turn. So let's look at the first 3 points here, starting with allergenicity. So the possibility that we might see an increase in the number of allergic reactions to food as a result of genetic engineering has quite a powerful appeal, because many of us have either experienced this problem before transgenic crops came about, or we know someone that has. However, there's no evidence so far that genetically engineered foods are more likely to cause allergic reactions than conventional foods. They've tested several dozen transgenic foods for allergenicity, and so far they've only uncovered a soybean that was never marketed, and the now famous Starlink corn. And even though the preliminary finding is that Starlink corn is probably not allergenic, the scientific debate still actually continues. Every year, some people discover that they have developed an allergy to a common food such as wheat or eggs, and some people may develop allergies to transgenic foods in the future. But currently, there's no evidence that transgenic foods pose more of a risk than conventional foods do. Let's now look at antibiotic horizontal transfer and antibiotic resistance together. So the use of antibiotic resistance markers in the development of transgenic crop plants has raised concerns about whether transgenic foods will play a part in our loss of ability to treat illnesses with antibiotic drugs. At several stages of the lab process, developers of transgenic crops use DNA that codes for resistance to certain antibiotics, and this DNA becomes a permanent feature of the final transgenic plant, even though it serves no purpose beyond the lab stage. So the question is, will transgenic foods contribute to existing problems with antibiotic resistance? And these existing problems are huge. One aspect of this topic is the risk of horizontal gene transfer. That is, transfer of DNA from one organism to another outside of the normal parent to offspring channel. Transfer of a resistance gene from transgenic food to microorganisms that normally inhabit our stomach and intestines, or transfer of resistance genes to bacteria that we ingest along with food, could help those microorganisms to survive an oral dose of antibiotic medicine during an infection. Now, although horizontal transfer of DNA does occur under natural circumstances and under lab conditions, it's actually quite rare in the acid environment of the human stomach. Another concern is that the enzyme product of the DNA might be produced at low levels in transgenic plant cells. While high processing temperatures would inactivate the enzyme in processed foods, ingestion of fresh or raw transgenic foods could result in the stomach containing a small amount of an enzyme that inactivates an orally administered dose of the antibiotic. This issue was actually raised during the approval processes for Calgene's Flavour Saver tomato and Cybergeigy's BT Corn 176. And in both cases, tests showed that orally administered antibiotics would remain effective. So while the risks from antibiotic resistance genes in transgenic plants appears to be low, steps are currently being taken to reduce the risk and to phase out the use of antibiotic resistance genes. Let's now consider the next 2 points here, starting with eating foreign DNA. So when scientists make a transgenic plant, they insert pieces of DNA that did not originally occur in that plant. And often these pieces of DNA come from entirely different species, such as viruses and bacteria. So is there any danger from eating this foreign DNA? Well, of course we eat DNA every time we eat a meal. DNA is the blueprint for life. All living things contain DNA in just about all of their cells. So what happens to this DNA? Most of it's broken down into more basic molecules when we digest a meal. A small amount is not broken down and can be either absorbed into the bloodstream or it's simply excreted in the feces. We suspect that the body's normal defence system eventually destroys this foreign DNA. Further research in this area would help to determine exactly how humans have managed to eat DNA for thousands of years without noticing any effects from the tiny bits that do sneak into the bloodstream. The cauliflower mosaic virus promoter. When scientists use transgenic technology to put a new gene into a plant, they put in additional pieces of DNA to direct the activity of that gene. One of these additional pieces of DNA is the promoter that turns the gene on. And the most widely used promoter is the cauliflower mosaic virus 35S promoter, often abbreviated as the CAMV 35S promoter, or simply the 35S promoter. This promoter was obtained from the virus that causes cauliflower mosaic disease in several vegetables, such as cauliflower, broccoli, cabbage and canola. There are concerns that the cauliflower mosaic virus promoter might be harmful if it were to invade our cells and turn on our genes. A multi-step chain of events would have to occur for the cauliflower mosaic virus promoter to escape the normal digestive breakdown process, penetrate a cell of the body, and insert itself into a human chromosome. While there have been no tests to determine whether this promoter has invaded human tissues, experiments with mice indicate that normal body defences eliminate these stray fragments of foreign DNA that do sneak into the bloodstream from the digestive tract. There is some evidence that this promoter poses very little threat, if any, to human health. People have been eating it in small quantities for hundreds of years. For example, when we eat vegetables that are infected with the disease. And although vegetables heavily infected with the cauliflower mosaic virus promoter are unappetising, there have been no documented negative effects on health from eating the virus or its promoter. Changed nutrient levels. How do genetically engineered foods compare with conventional foods in nutritional quality? This is an important issue and one for which there will probably be a lot of research in the future, as crops that are engineered specifically for improved nutritional quality are marketed. However, there's only been a few studies to date comparing the nutritional quality of genetically modified foods to their unmodified counterparts. The central question for genetically engineered crops that are currently available is whether plant breeders have accidentally changed the nutritional components that we associate with conventional cultivars of a crop. So, for example, because isoflavones are thought to play a role in preventing heart disease, breast cancer, and osteoporosis, the isoflavone content of Roundup Ready soybeans has been investigated by several researchers. The studies completed so far do not resolve the issue of whether Roundup Ready soybeans have isoflavone levels comparable to conventional varieties. but the differences that were found in experiments appear to be small or moderate in comparison with natural variation in isoflavone levels. Additional evidence may clarify the arguments for and against Roundup applications as a risk factor in soybean cultivation. Industry studies submitted in support of applications for permission to sell transgenic crops indicate that the nutritional components that are commonly tested are similar in transgenic foods and conventional foods. Our next group of risks and concerns involves damage to the natural environment. For example, we have crop-to-weed gene flow, antibiotic resistance, leakage of GM proteins into the soil, and reductions in pesticide spraying. Are they real? Let's look at each of these in turn. The suggestion that Bt corn pollen might kill the monarch butterfly larvae galvanised public interest in the effect of transgenic crops on the environment. So let's think about crop-to-weed gene flow. Hybridisation of crops with nearby weeds may enable weeds to acquire traits that we wish they didn't have, such as resistance to herbicides. And research results indicate that crop traits may escape from cultivation and persist for many years in wild populations. Genes that provide a competitive edge to weeds, such as resistance to viral disease, could benefit weed populations around a crop field. Many cultivated crops have sexually compatible wild relatives with which they can hybridise under favourable conditions. And the likelihood that transgenes will spread can be different for each crop in each part of the world. For example, there are no wild relatives of corn in the United States or in Europe for transgenic corn to pollinate, but these wild relatives do exist in Mexico. Soybeans and wheat are self-pollinating crops, so the risk of transgenic pollen moving to nearby weeds is very, very small. However, that small risk must be balanced against the fact that there are wild relatives of wheat in the United States. Next, we consider antibiotic resistance and leakage of GM proteins into the soil. So there's also concern that transgenic plants growing in the field will transfer their antibiotic resistance genes to soil microorganisms, thus causing a general increase in the level of antibiotic resistance in the environment. However, many soil organisms have naturally occurring resistance as a defence against other organisms that generate antibiotics. So genes contributed occasionally by transgenic plants are highly unlikely to cause a change in the existing level of antibiotic resistance in the environment. Many plants leak chemical compounds into the soil through their roots. So there's concerns here that transgenic plants may leak different compounds compared to conventional plants. And this is as an unintended consequence of their changed DNA. Speculation that this may be happening leads to concern about whether the communities of microorganisms living near the transgenic plants may be affected. The interaction between plants and soil microorganisms is very complex, with the microorganisms that live around plant roots also leaking chemical compounds into the soil. So a lot more research has to be done before we understand the relationships that occur between microorganisms and conventional crops. Attempts to discover whether transgenic plants are changing the soil environment and whether they're changing it in good ways or bad ways are actually hindered very much by our lack of basic scientific knowledge. Reductions in pesticide spraying, are they real? One of the most appealing arguments in favour of transgenic plants is the potential for reducing the damage we do to the environment with conventional methods of farming. Pest-resistant crops such as Bt corn and Bt cotton have been widely promoted as a means to reduce the spraying of pesticides, while herbicide-tolerant crops such as Roundup Ready soybeans are said to reduce the application of herbicides. Large reductions in chemical spraying have been claimed to result from the introduction of these transgenic varieties. Are the claims true? Bt cotton is the only crop for which claims of reduced spraying are clear. Analysts painted a mixed picture on the results of planting Roundup Ready soybeans. Bt corn and herbicide-tolerant cotton and corn have not resulted in clear reductions in the spraying of chemicals. Finally, we look at disruption of current practices of farming and food production in developed countries. Crop-to-crop gene flow. Hybridisation of transgenic crops with nearby conventional crops raises concerns about separation distances to ensure purity of crops and about who has to pay if unwanted genes move into a neighbour's crop. As identity preservation and segregation of GM from non-GM crops become factors in marketing products, it's obviously of great importance to ensure that hybridisation is not occurring in the field. And there are many factors that influence the potential for gene flow from crop to crop. Some crops are highly outcrossing, with pollen carried to other fields by wind and by insects. Other species are highly self-pollinating, with very little potential for pollen transfer to neighbouring plants. And because of the differences among crop species, every case must be evaluated individually for potential to contribute to gene flow from transgenic to conventional crops. If GM pollen pollinates plants in a neighbouring field, then the issue of genetic trespass may arise. What level of GM presence, if any, should be allowed in products that are sold as organic or conventional? Should GM farmers and companies bear the responsibility for preventing gene flow, or should conventional and organic farmers pay to protect their products from gene flow? Should GM versions of outcrossing plants be banned as simply being too risky, while GM versions of self-pollinating plants are permitted? These issues have already prompted several lawsuits, and they'll continue to be a factor in the development and use of transgenic plants for years to come.